Hello, my dear students. Welcome to IKSR chemistry classes. In this video, we will discuss biological oxygen demand, that is BOD, and its experimental determination. It is a very important topic under a skill development program, and questions related to it are asked frequently in various general as well as competitive examinations. Let us first of all introduce the chapter. The microorganisms such as bacteria are responsible for decomposing organic wastes such as dead plants, leaves, manure, sewage, or food wastes present in a water sample. These are dead plants. It is a plant leaf present in water. The manure used for the growth of plants, and it is sewage, municipal sewage, and waste foods. If these materials are present in a water sample, these are decomposed by microorganisms such as bacteria. When such a water sample is allowed to stand, the aerobic bacteria begin to process the breaking down of these organic wastes and consume the dissolved oxygen. When a water sample containing organic wastes discussed earlier, the bacteria start to decompose these organic wastes, and during this process bacteria consume the oxygen dissolved in water. Such bacteria which acts during the decomposition of organic wastes by using oxygen are called aerobic bacteria. As a result of this process of breaking down organic wastes present in the water sample by aerobic bacteria, the available dissolved oxygen is consumed by aerobic bacteria during this process. Thus, other aquatic organisms become deprived of available dissolved oxygen because a portion of dissolved oxygen has been consumed by aerobic bacteria during the decomposition of organic wastes. It affects the aquatic life of water body adversely. What is dissolved oxygen? What is biological oxygen demand? Let us see what is biological oxygen demand, which is abbreviated as BOD. The amount of oxygen, the amount of dissolved oxygen required by aerobic biological organisms. Let us see the amount of dissolved oxygen required by aerobic biological organisms such as aerobic bacteria in a body of water or waste water sample. To break down organic materials present in the given volume of water sample at a certain temperature over a specified period of time is called biological oxygen demand of the given water sample. Let us see it again. The amount of dissolved oxygen required by aerobic biological organisms in a water body or wastewater sample to break down organic materials present in it in the given volume of water sample. The volume of water sample should be specified at a certain temperature. The temperature also must be specified over a specific period of time is called biological oxygen demand. Three things must be specified in the definition of biological oxygen demand: the volume of water sample, the temperature, and the period of time. How biological oxygen demand is expressed. The BOD is generally expressed in milligrams of oxygen consumed. How many milligrams of oxygen are consumed per liter of water sample during 5 days of incubation at 20 degrees centigrade. The temperature should be 20 degrees centigrade. The time span should be 5 days. The volume of water body should be in liter, and the amount of oxygen consumed should be in milligrams. The biological oxygen demand of a given water sample is expressed in terms of milligrams of oxygen consumed per liter of water sample during 5 days at an incubation temperature of 20 degrees centigrade. The volume of water sample should be in liter. The time should be 5 days. Temperature should be 20 degrees centigrade. Volume should be 1 liter. Then the milligrams of oxygen consumed by aerobic bacteria during the breakdown of organic wastes is called biological oxygen demand. BOD as a scale of water quality. A sufficient concentration of dissolved oxygen is essential for maintaining the aquatic life in a water body. The dissolved oxygen should be proper for the maintaining aquatic ecosystem in a water body. It means the greater the biological oxygen demand of a water sample, the dissolved oxygen will be less available to other aquatic organisms. If the biological oxygen demand of a water sample is higher, the dissolved oxygen available to other aquatic organisms will be lower. The BOD of a fresh water body should be less than 5 ppm. If the BOD value of a water sample is greater than 17, it is highly polluted. Significance of BOD value. The BOD value is the measure of organic pollutants present in aquatic ecosystem. greater the BOD value of a water system, means there are greater amount of organic pollutants present in the aquatic ecosystem. A high value of BOD indicates that the amount of dissolved oxygen available for other aquatic organisms such as fish is low. The concept of BOD is used in sewage treatment or wastewater treatment to destroy and decay the organic wastes through the aerobic organisms. Thus, it is very useful in water pollution control management. The knowledge of BOD value of a water sample is important in the water pollution control management. The concept of BOD is helpful in evaluating self-purification capacities of water bodies such as streams. Now, the main portion of this video is determination of BOD. Let us see the principle of this process. The BOD test. The evaluation of BOD value of a water sample consists in taking the given sample in a suitable concentration in distilled water in 2 BOD bottles. The given water sample is taken in suitable concentration in distilled water in 2 biological oxygen demand bottles. One bottle is incubated in a BOD incubator for 5 days at 20 degrees centigrade, while the dissolved oxygen, initial dissolved oxygen content D1 in the second bottle is determined immediately. In second bottle, the dissolved oxygen level, initial dissolved oxygen level D1 is determined immediately after sampling. At the end of 5 days, the dissolved oxygen content, that is D2, in the incubated bottle is determined. The water sample in suitable concentration is taken in 2 BOD bottles. In 1 BOD bottle, the initial dissolved oxygen content D1 is determined immediately, while the other BOD bottle containing water sample in suitable concentration is incubated in a BOD incubator for 5 days and at 20 degrees centigrade, and at the end of 5 days the dissolved oxygen content D2 in this bottle is determined. Then the BOD, biological oxygen demand, is calculated using this relationship D1 minus D2 by P ppm. Here P is decimal fraction of water sample used. Decimal fraction of the sample used is P. It means P is volume of water sample divided by volume of diluted solution. The water sample has volume, certain volume, and it is diluted with the help of distilled water to another value. Initial volume of water sample divided by volume of diluted solution is equal to P, which is called decimal fraction of the sample used. D1 is dissolved oxygen of diluted sample in milligram per liter immediately after the preparation. D1 is initial dissolved oxygen level in the diluted sample in milligram per liter just after the preparation of sample, while D2 is dissolved oxygen level of diluted sample in milligram per liter at the end of 5 days of incubation at 20 degrees centigrade. This is final D2. This is final dissolved oxygen level in milligram per liter, while D1 is initial dissolved oxygen level in the same unit, that is milligram per liter. The following apparatus are required during the determination of biological oxygen demand. BOD bottles of 300 ml capacity. 2 such bottles are required. BOD incubator, in which the BOD bottle containing water sample in suitable concentration is placed at 20 degrees centigrade for 5 days. It is burette stand, conical flask, pipette, measuring cylinder, and measuring flask. These apparatus are required during the experimental determination of BOD. Reagents required. First of all, we need distilled water to dilute the given water sample. Phosphate buffer solution. For the preparation of this solution, 8.5 gram potassium dihydrogen phosphate, 21.7 gram potassium monohydrogen phosphate, and 33.4 gram sodium monohydrogen phosphate heptahydrate and 1.7 gram ammonium chloride are dissolved in 500 ml distilled water, and then the volume of the solution is made 1 liter by adding further amount of distilled water. The pH value of this phosphate buffer solution is around 7.2. The another reagent required for the determination of BOD is magnesium sulfate solution. For its preparation, 22.5 gram magnesium sulfate heptahydrate is dissolved in distilled water and the volume is made 1 liter. The another reagent is calcium chloride solution. For its preparation, 22.7 gram anhydrous CaCl2 is dissolved in distilled water and the volume is made 1 liter. Another solution required is ferric chloride solution. 0.25 gram FeCl3 6H2O is dissolved in distilled water and the volume is made 1 liter. And reagents required for determination of dissolved oxygen are required. It has been discussed in the previous video. And now let us see the procedure for the determination of BOD of a water sample. 10 ml of water sample taken from a water body is taken in a 1 liter measuring flask. 100 ml of the water sample is taken in a 1 liter measuring flask. 1 ml each of phosphate buffer, magnesium sulfate solution, calcium chloride solution and ferric chloride solution are added to this water sample to provide nutrition to microbes. Then the volume is made 1 liter by adding distilled water. 10 ml water sample is taken in a 1 liter measuring flask and 1 ml of these solutions: phosphate buffer solution, magnesium sulfate solution, calcium chloride solution and ferric chloride solution each are taken in the measuring flask, and then the volume is made 1 liter by adding distilled water. This solution is taken into two 300 ml BOD bottles, one for incubation at 20 degrees centigrade for 5 days and the other for determination of initial dissolved oxygen in the mixture. This diluted water sample containing nutrients for microbes is taken in two 300 ml BOD bottles. One BOD bottle is incubated at 20 degrees centigrade for 5 days and the other determined the initial dissolved oxygen D1 content of one BOD flask by Winkler method. In first BOD bottle, the initial dissolved oxygen level D1 is determined with the help of Winkler method as discussed in the previous video. The dissolved oxygen D2 content in the incubated bottle after the end of 5 days is determined with the help of same method, and the results of two experiments are noted down. The BOD of the given sample is calculated using the relationship given earlier. BOD is equal to D1 minus D2 by 0.01 ppm. Here P is equal to 10 ml initial volume of the water sample divided by volume of diluted water sample. 10 ml was the initial volume of the water sample, while the volume of diluted solution was 1000 ml, and therefore the value of P is equal to 0.01, which has been taken here. Now the time for self-assessment. In a BOD determination, 600 ml waste water is mixed with 294 ml distilled water in 300 ml BOD bottle. The BOD of this solution was initially 9.1 ppm, and after 5 days of incubation at 20 degrees centigrade, the final dissolved oxygen, the initial dissolved oxygen was 9.1, while at the end of 5 days incubation at 25 degrees centigrade, the dissolved oxygen level was 2.8. Then calculate the BOD of the water sample. This should be DO, not BOD. The DO of the solution. BOD should be read as DO. The dissolved oxygen of this solution was initially 9.1 ppm, while at the end of 5 days of incubation at 20 degrees centigrade, the final dissolved oxygen level was 2.8 ppm. Then we have to calculate the DO value of waste water sample. And it is the solution. Here P is equal to 6 ml volume of the water sample divided by 300 ml volume of the diluted water sample, and on solving it we have P is equal to 0.02 and BOD is equal to D1 minus D2 by P ppm. D1 is 9.1 as given in the problem and D2 is 6.8. Let us see in the previous slide. This should be 6.8. This should be read as 6.8, not 2.8. 6 divided by 300 ml, that is 0.2 is the value of P. BOD will be equal to D1 minus D2 by P ppm. D1 is 9.1, D2 is 6.8 by P, and the value of P as already has been calculated 0.02, and therefore the BOD value is 2.3 divided by 0.02 ppm. And on solving it we have BOD value 115 ppm. Let us see, it is a quite large value. It is a quite large value, and therefore the water body from which the water sample has been taken is highly polluted, is highly loaded with organic wastes, and this is why the BOD value of this water sample is very high. Thank you for watching the video with patience. Please like and share it. If you have not subscribed my channel yet, please subscribe it so that you may get information regarding my new videos in time. Thank you again.